So before moving on to the 3-point crosses, we'll just have a brief idea about another terminology that is associated with linkage. The terminology is linkage ratio. Now, what do we mean by linkage ratio? So, by definition, if complete linkage exists between two genes and the organism are heterozygous at both loci and mated, a unique F2 ratio results, which we designate as the linkage ratio. So let us illustrate this phenomenon with an example. The example is that of Drosophila. Here Drosophila, two flies are taken. The two genes in consideration are H, which is heavy wing veins, which results in heavy wing veins, and BW, this is eye color gene, which results in brown eyes. The wild type allele, H plus, results in normal thin wings, and the wild type allele BW plus results in red eye color of the flies. So these are the two genes taken in consideration to exemplify the phenomena of linkage ratio. Now let us look at the cross. So that is a cross. So as you can see, we have a parent that is normal thin wing veins, brown eyes, the first parent. So it's thin veins, brown eyes, and this is the heavy veins, red eyes. Now let's elaborate the diagram. So we have thin veins, brown eyes, heavy veins, red eyes. Now we'll be getting gametes from both these flies. So let us look into the gametes. So we'll be obtaining one gamete, which is the heavy thin veins, brown eyes, and one gamete from here, which will be the heavy veins and red eyes. Now we have a F1. So the F1 is a heterozygote. One chromosome is coming from the mother and the other is coming from the father. So basically this is an autosome. Hence the F1 is not denoted as a male or a female, as the genes concerned are autosomal genes. Now in the F1, as I said, each fly receives one chromosome from each pair of parent. All flies are heterozygous and they exhibit the dominant traits or the wild type traits, thin vein and red eyes. Now, when these F1 flies are test crossed, so when this F1 is interbred, each F1, one thing is that when this F1 flies in this particular picture is test crossed with the parent, so this test crossed with the recessive parent, as you can see, since the genes are linked, so the F1 will produce only two kinds of gametes. There is no recombination between the two genes under study, as the two genes under study are linked. So we will be getting only the parental gametes from F1, and this is the other parent. This will produce only one kind of gamete. Now we have two situations. So the first situation is when F1 is interbred with another F1. So these are the parentals gamete. These are also the parental gametes. When they are inbred, we are getting a product like this. What does the product signify? It is 1/4 thin wing veins, brown eyes, half thin veins, red eyes. That is the recombinant category, and 1/4 heavy wing veins and red eyes. So the ratio of 1 heavy is to 2 wild type is to 1 brown. This is 1:2:1 ratio is a characteristic of complete linkage. If we see another cross where the F1 is test crossed with a parent, you can see that we are obtaining only two kinds of progeny. The ratio is 1:1, thin brown and heavy red. And the ratio, as you can see, these two progenies are only the parental progeny. There is no recombinant progeny obtained in such case. So this is a typical example when what happens when linked genes are crossed. Either we saw two types of crosses where the two linked genes in concern were the wing vein gene and the eye color gene. And we saw the heterozygous F1 being interbred with another F1, giving a perfect complete linkage ratio of 1:2:1, one parental, two recombinant type, one another parental type. And we also saw the F1 being test crossed with another parent. And again, in this case, the phenotype that we observed were only the parental categories. Thus, we can assume that when large number of mutant genes in any given species are investigated, genes located on the same chromosome show evidence of linkage to one another. As a result, linkage group can be identified, one for each chromosome. So in this particular case, the wing vein gene and the color gene are showing a linkage group. So that was the last part of another definition associated with linkage and complete linkage. And finally, just to summarize what we have done till now, that is the detection of linkage. Just always remember, I'm not getting into the details of the crosses because we have discussed the crosses a number of times. Remember the ratio, this is important. So if the ratio is 1:2, you're getting 4 categories of phenotypes and 2 categories showing parental is similar to the parents, and 2 categories are recombinants, and they are observed in the ratio 1:1:1:1, then we will safely assume that the 2 genes are not linked, they are being independently assorted. But the moment we are considering 2 genes and we are getting a ratio of 1:1, that is only 2 genotypes are observed, and that too the 2 genotypes in concern are the parental genotypes only, then we will safely assume that the 2 genes under study are completely linked. Finally, if there are 2 genes and we get 4 categories of phenotypes, so there are 4 phenotypes, 2 parental type and 2 recombinant type, but the ratio of the parental is much, much greater than that of the recombinants, we will safely assume that the 2 genes in concern are incompletely linked. So with that, we now move on to the next part of our class, that is the 3-point cross. So when we will be talking about 3-point cross, it was Alfred Sturtevant, a student of Morgan in 1913, determined the fact that the recombination frequencies could be used as a measure to measure the genetic distance between 2 genes on a genetic map. So the amount of recombination could give a reflection about the distance between the 2 genes present on a chromosome. And the genetic distance is measured in map units, where one map unit is defined as the interval in which 1% crossing over takes place. So if we have 1% crossing over, that is equivalent to the fact that there is 1 map unit distance between the 2 genes. The map unit is also referred to as centimorgan, a term named by Sturtevant in honor of Morgan. And finally, we should all keep in mind that the crossover frequency is not same as the recombination frequency. The crossover frequency means it is the frequency of physical exchanges between chromosomes in meiosis for the region between the genes. So it's the frequency of physical exchange formed by the formation of chiasma, while the recombination frequency refers to the frequency of recombination occurring between genetic markers in a cross. What do we mean by genetic markers? Genetic marker means certain genes which will give us a very prominent phenotype. For example, the one we just now studied in case of linkage group, the wing vein and the eye color. So remember that the crossover frequency is the frequency of physical exchange between chromosome. The recombination frequency is the frequency of recombination between genetic markers occurring in a cross. So with that, we start with a beautiful example. Now let us look at this particular example. So continuing. So let us assume that there is a plant which has yellow, elongated, dry fruits, and the other plant is producing purple, round, juicy fruits. In this particular case, as it is evident, P plus dominant to P, R plus dominant to R, J plus dominant to J. So this is evident. And another important thing to remember is that we are having a combination of P plus, R plus, J plus. Consider this plant. This is a homozygous recessive with P, R, J, right? So what other gametes can we obtain? A crossing over might take place here. So first category are the parental gametes, P plus, R plus, J plus. And the second category is another parental gamete, P, R, J. So these are the parentals. But now since there are 3 genes present, the first crossing over is happening here. So what are the combinations we can get from here? P plus, R, J. So this chromosome is becoming this. And P, R plus, J plus. And this is becoming this. So this might be one probability, or we might have another probability. If instead of crossing over here, we have a crossing over here. So in that case, what are the combinations we will get? We will get a combination of P plus, R plus, J, and P, this is J, R, J plus. So we'll be getting 1 gamete, 2 gamete, 3, 4, 5, 6. So there are 6 possible gametes. There are 6 kinds of gametes possible from this particular picture. One thing to keep in mind is that in this kind of problems, we are not sure that which is the middle gene. Given in this picture, based on that picture, I have drawn this diagram considering the fact that R is the middle gene. We are considering 3 genes and assuming that R is the middle gene, I drew these 6 kinds of gametes. So when this kind of cross will be given, a data will come with it. And the data reflects that these 2 plants were crossed and 8 categories of progenies were obtained. The 8 categories of progenies are obtained. How is it possible? Because I've drawn 6 categories of gametes. So 6 categories of progenies are possible that you people have understood. But what are the 7th and the 8th category of gametes? But before coming to that, so these are the parental ones. Remember, since the heterozygous is being crossed with a homozygous recessive, so this plant will always contribute to nothing else but P, R, and J. And P, R, and J are always recessive. So the first two categories of gametes we are seeing are the parentals, right? So we have the two parental progenies. Then if you remember, I said that let us assume there was a cross between P and R, crossover between P and R. Then we will observe these two category of gametes. So these two category of gametes are also being observed in this case. So these are the parents. Okay. So these two category of gametes are also being observed in this case. Now comes a situation where my drawing of the gametes is not matching with the phenotype that is being observed. If you here see, if you see that there is a crossing over between R and J, then in that case, this situation shouldn't arise. We should get a gamete condition like this. But this gamete scenario is not matching to this one. And finally, we have another scenario of which the gametes I have not drawn. So immediately there arises a question that whether R is the middle gene or not, because these gametes of mine are not matching. And again, how did we get the last two category of gametes? So talking about the last two category of gametes, let me erase all in. Let me draw it once more. So we have P plus, R plus, J plus, P, R, J. So one crossing over took place here. That gave rise to P plus R, J and P, R plus J plus. That was case one. Case two might be that a second event of crossing over, independent of the first is not occurring. They're not two occurring, they're not occurring simultaneously. The exclusive events is occurring here. So the gamete should have been PR plus J and PRJ plus. Our gamete is not matching. And the final crossover that can take place in cases like this, where we have three genes in concern, P plus R plus J plus, PRJ, is that simultaneously two events of crossing over are occurring between the two sets of genes. So there is a crossover event occurring between P and R, and at the same time, a crossover event is occurring between R and J. In that case, we will be getting a combination of P, R plus J and P, R, J. So let me change the color of the. So this is P, R, P plus R, J plus, and P, R plus J. From this particular picture also, if you look at the double crossovers, what we are getting, P plus R, J plus. P plus R, J, no, our double crossovers are also not matching. This is known as a single crossover SCO. This is also known as a single crossover SCO. So we can name it as SCO1. We can name it as a SCO2. And this is referred to as a DCO. From the diagram I have drawn here, just a second. From the diagram that I've drawn here, as you can see, this is a DCO. The SCO1 gametes are matching, but our SCO2 gametes, PR plus J and PRJ plus, is not matching. Neither is a double crossovers matching. So it might be that R is not the middle gene. In a data like this, how to analyze the data? The maximum number of progeny are of the parental types. So, and the minimum number of progeny are of the double crossover types, because you can imagine that simultaneous crossover event occurring between three genes which are sitting adjacent to each other is basically a rare phenomenon. And because this is a rare phenomena, so it is a fact that these progenies will be the least in number. So whenever we have a data like this, the first thing we need to identify is the parental class and the double crossover class. So from the data, now let's start with the data. We have identified the parental class and we have identified the double crossover class. Now, as I said, so if the R is the middle gene, then this is how I have the parents are drawn. The region between P and R is region 1. Any crossover event taking place between region 1, this is known as a single crossover event. Again, the region between R and J is known as region 2. So crossover event occurring between R and J is known as another, again, single crossover event, and that is SCO2. And finally, as I said, what is the DCO? The DCO is a crossing over event occurring here and a crossing over event occurring here. Now, I have showed you the single crossover events. I've showed you the two categories of two types of single crossover 1 and single crossover 2, and I've showed you the DCO. And from that above analysis, we understood that the DCO is not matching with the data given here if we consider R as the middle gene. So how do we understand which is the middle gene? Now look at the parents, P plus, R plus, J plus. This is one. The other one is P, R, J. Now let us look at the double crossovers. P plus, R plus, J, P, R, J plus. We have to seek that gene which has changed its position with respect to the other two genes. Once more, we have to seek that gene which has changed its position with respect to the other two genes. You see, in this scenario, the parents P and R, P plus and R plus are together. See, similarity, P plus and R plus together. PR together, PR together. It is the J gene which has changed its position with respect to the parents. Hence, this is not the correct picture. J is supposed to be the middle gene. So if we analyze this diagram once more, this will be P plus, J plus, R plus, P, J, R. So this is how, first we have to, in a problem given like this, first we have to understand the correct order of the genes. The moment we understand the correct order of the genes and we understand which is the middle gene, then we will rearrange the data as per the correct order of the genes. When we have understood how to identify the middle gene, now let us look at this were the single crossover region. So this is what happened. This is a correct gene order, and this is the double crossover. So you can see P plus, J, R plus. So P plus, J, R plus. Here the order is not arranged properly. And the other one is P, J plus R, P, J plus R. So these were the parentals. P plus J plus, sorry. These both are not the parentals. This is one parental. This is one parental. See, now you see, match it up with the main data. So the two parentals, this is one DCO, P plus J R plus, we have one DCO. And this is the other DCO, P, J plus R. So once we have successfully identified the middle gene and have arranged our genes in the correct order, preferably what is done is that we rearrange the data. So we have rearranged the data keeping G as the middle gene. So these are the parentals. Now we have, remember, recombination between region 1, SCO1. So P plus so SCO1 will be recombination between this point. So P plus JR, P plus JR, and the second one will be P, J plus R, J plus R plus. So P, J plus R plus. So this is the crossing over event occurring between P and J, that is region 1. So they are referred to as single crossover region 1, their recombinants. And the second crossover event that will take place will be in region 2. So when we are having a second crossover in the region 2, so we'll be having P plus J plus R, P plus J plus R, this is a typological error, and P, J, R plus, P, J, R plus. So single crossover in region 2. And finally, the double crossovers, I showed you that the double crossovers will be P, J, R plus and P, J plus R. Now the problem part. The problem is to find what are we supposed to do with the data. That we have to calculate the distances between the P, J and R gene. And that is why these values are given. So now what we do is that we rearrange them in a table. So these are my parental. This is my single crossover region 1, single crossover region 2, double crossover. And this is total is 500. So to obtain the amount of recombination, amount of progeny or the percent of progeny present per category, what do we do? The amount of parental is 0.7. We add up the parentals, divided it by the total number of progeny. The amount of single crossovers is 98 by 500, that is 0.196 in region 1. The amount of single crossovers in region 2 is, add up the total divided by 500, that is 0.088. And finally, the amount of double crossovers is 0.012, where 6 double crossovers are present in 500. So we get the amount of the frequency of each category present. From that, we use the formula. This is the standard formula. Distance between P and J. So it is it's the number of SCO1 plus the DCO. The number of SCO was 98. DCO is 6 divided by 500. You get the frequency. If you want to convert it into percentage, multiply it by 100, is 20.8. Distance between J and R, same. The single crossovers plus the double crossovers divided by the total progeny. You get the frequency, multiply it into 100, you get the percentage. And similarly, distance between P and R. Now remember the position of the genes. We calculate the distance between P and J. We have calculated the distance between P and J and R. So adding up these two values will simply give you the distance between P and R, the two genes at the ends. Or otherwise, you can put it in the formula, single crossovers region 1 plus single crossovers region 2 plus twice double crossovers, divided by the total progeny into 100. So this is how we get the distance between P and J J and R, and PR is a addition of P and J and R, or you can put it in the formula and calculate. So this was distance between P and J, distance between J and R, distance between P and R, and finally on the map, this was how we do it in the map. Remember, in the map, if you do not write the unit, marks will be deducted for not writing the unit. You have to draw the map in such a manner so that it is, from visualization, it can be understood that the distance between P and J is almost double that of J and R. Distance between J and R is only 10 map unit. The distance between P and J is 20.8 map units. It's almost double. So you can see, if you look at the visualization also, this distance is almost double than this distance. And finally, one thing that has not been written here, the distance between P and R, that should also be mentioned in the map. That is 30.8 map unit. So this is how the map is drawn. And there is an addition to this part, that is that we have to calculate two values. One is the interference and other is the coefficient of coincidence. Now, to calculate interference and coefficient of coincidence, we need to have another value which is referred to as the observed DCO. So how do we calculate the observed DCO? The observed DCO is calculated by adding recombination frequency region 1 divided by recombination frequency region 2. So that is the observed DCO that you have observed in the cross. Sorry, this is not the observed DCO. Very sorry. This is the expected DCO. Sorry. Observed DCO is in the cross. You saw there were 6 DCOs on 500 progenies. I've written, this is written wrong here. So this is observed DCO. But we need to have, so that is observed. The amount of double crossovers present in the data, that is the observed DCO. But we need to have the expected DCO. That means if crossing over in region 1 is independent of crossing over in region 2, assuming that the crossing over occurring between region 1 is totally independent. If you remember, P, R, J, P plus, R plus, J plus. So whenever we are having a double crossover event, so there is a crossing over occurring between P and R and J. So the concept of expected DCO is given by the fact that the crossing over between P and R is independent with respect to the crossing over occurring between R and J. If such a case arises, then the probability of double crossovers in the 2 regions is equal to the probability of the product of the probabilities of the 2 events. So recombination frequency of region 1 divided by 100 into recombination frequency of region 2 divided by 100 will give you the expected DCO. So if you remember the recombination frequency, the data was, if you remember this data, let's go back to the data. The recombination frequency between region 1, that is 20.8% or 0.208. And the recombination frequency between region 2, that is 10 map units, it's 0.010. So when we are calculating the expected DCO, we have to keep it in mind. So it's 0.208, 0.110, 0.100. So 0.208 is your expected double crossover. So once we have obtained the expected DCO, now there are 2 terms to be understood. One term is interference and the other term is coefficient of coincidence. So there is this interference and there is this coefficient of coincidence. Again, let us draw this scenario of the cross we studied P plus R plus J plus. J is our middle gene. So considering that, we'll draw the with P plus, J plus, R plus, and P, J, R. Whenever there is an incident of a double crossover event, so crossing over is occurring between P and J, and at that given moment, crossing over is occurring between J and R. We just now understood that we calculate the expected DCO considering that this crossover event is independent of that crossover event. But in reality, if three genes are sitting adjacent to each other, then chiasma formation and crossing over between two genes tends to interfere with the chiasma formation and crossing over between the other two genes. Because if there is a chiasma formation and crossing over event occurring here, this phenomena will have a tendency so this phenomena will have a tendency to interfere with this second crossing over occurring between J and R, because remember, in both the crossing overs, J is being involved. So whenever there is this tendency to interfere with this crossing over, this is known as interference. So the first crossing over event will have a tendency to interfere with the second crossover event when two crossing overs are occurring simultaneously between three genes which are sitting adjacent to each other. Thus, keeping in mind this view, now we can say that the presence of one crossover interferes with the formation of another crossover nearby, and this phenomena is called interference. The extent of interference is expressed as coefficient of coincidence. That is, how do we calculate the coefficient of coincidence? We have the observed double crossover. The observed double crossover for us in this particular example was 6, if you remember, on 500. And we calculate the expected double crossover. I told you how to calculate the expected double crossover. So observed double crossover divided by expected double crossover will give you a value. So let us suppose this value is X. So the coefficient of coincidence in this particular example is X. So now we calculate interference 1 minus X. So that is how we calculate interference and coefficient of coincidence. If it so happens that the value of coefficient of coincidence is 1, that means the frequency of observed double crossover is exactly equal to the frequency of expected double crossover. That again means that the crossing over is occurring, crossing over of one region is becoming totally independent of another region. When the coefficient of coincidence is 1, we have to assume that the crossing over of one region is becoming exactly independent of another region. In such a condition, if coefficient of coincidence is 1, then interference becomes 0. So one crossing over is not at all interfering with the crossing over event taking place simultaneously in the nearby place. And what might be the other situation? The other situation is that if the coefficient of coincidence is 0, that means if such a situation arises, so if we place a value 0 here, so you can see the interference is 1. So if there is an interference 1 and the coefficient is 0, then we are having an interference value of 1. That means that one crossing over is totally interfering with the happening of the other crossing over. And so the other crossing over is not taking place. So we will not be getting double crossover phenotypes in such a scenario. See, double crossover, how do we get double crossovers? We get double crossovers because two crossover events are occurring simultaneously at 2 points. Then only we get a double crossover phenotype. But if interference is 1, that means this crossover event is totally preventing the happening of this event. So this event cannot occur as a result of that crossover. If such a situation arises, then there will be no double crossovers. So keep this in mind. Because in problems, what you will see is that as we started this problem with 8 phenotypes, 2 parentals, 4 recombinants, 2 double crossovers. But in some problems, you will see there are 6 phenotypes, 2 parentals, 4 recombinants, no DCO. No DCO means that interference is 1. So one crossing over event is totally interfering with the happening of the other crossover event. And finally, just summarizing what we understood, that remember this, double crossovers are rare. In a given data, they are the least to occur. They are not often as expected as the observed rate of single crossovers. Second point to be noted is that whenever there's a crossing over occurring between 2 genes and there's a chiasma formation, then there's a tendency that another crossover event will not occur. So if it occurs, it will occur at a very low rate. So there will be a tendency of interference. When interference is 1, that is total interference, there will be no crossing over occurring in the other region. Now, the coefficient of coincidence expresses the extent of interference. The interference and coefficient of coincidence are inversely related, as I just now explained. The value of 1 means the number of double crossovers that occur is what to be predicted on the basis of 2 independent events, and there is no interference. So when we have a CC value of 1, there is no interference. But when we have a coefficient of coincidence value 0, that means there are no DCOs, double crossovers, because one crossover event is totally interfering with the formation of another double crossover event nearby. So interference is total, and in those cases will not get a double crossover phenotype. So we'll stop here today.